Hello everyone and welcome. In this video I'll be explaining all the answers and questions for May June 2018 Paper 1 Variant 19700. Of course there are timestamps for any specific questions that you would like to check out. Let's get started. Now, which statements about resolution and magnification are correct? So here would most likely be looking at definitions. Resolution is the ability to distinguish between 2 points or 2 separate objects that are very close together. This is a correct definition. And we also have magnification, which is the number of times larger an image is compared with the real size of the object. Another correct definition, so we could immediately identify the answer as A. Next is an eyepiece article has a scale with 100 divisions. A stage micrometer has a scale with 50 divisions, each of which is 0.40 millimeters apart. Using a times 40 objective lens, the whole length of the stage micrometer scale lines up with the 15 divisions of the eyepiece article, meaning that the 50 divisions will line up against 15 divisions when we use this, which is not really important for this question. What is the actual length of 100 division scales, 100 division scale of the eyepiece article? So let's begin by saying that 15 divisions of the eyepiece article will be lining up against 50 divisions of the stage micrometer. How many stage micrometer units? I know that they're not more, but let's see how many could possibly line up. That will be 333.3. Now we understand that 50 basically has a length, 50 divisions will have a length of 50 times 0.04. Because a single eyepiece article unit will have, a single stage micrometer unit will have a length of 0.04, then basically 0.04 times 50 will be a length of 2. That's the total length of the 50 divisions. This length will also be multiplied by 0.04, and that should be the length of the eyepiece article, which happens to equal to 13.3 millimeters. In other words, 13 millimeters, which is B. He says that a prokaryotic cell which is 1 micrometer in diameter is magnified 50,000 times in an electron micrograph. What is the diameter of the cell in the electron micrograph? Okay, so we want to look for the image length, knowing the actual length. We already know this triangle. It says that image length is at the top, magnification and actual length are at the bottom. And if I want to find the image length, then I'll have to multiply the magnification by the actual length, meaning 1 multiplied by 50,000 will equal to 50,000 micrometers, which is not an option here. Instead, we have all the sizes in millimeters. In order to convert a micrometers to millimeters, you have to divide by 1,000. So from micrometer to millimeter, divide by 1,000, and if it's the opposite case, then multiply by 1,000. I have mentioned it's 50,000 micrometers. Now divide this by 1,000 and you should get 50 micrometers, which is exactly 5 by 10 to the power of 1, millimeters, which happens to be C. Next question. A scientist carried out an experiment to separate cell structures in animal cells. The cells were broken open to release the cell structures, and the extract was filtered into a centrifuge tube and then spun in the centrifuge. The heaviest cell structure sank to the bottom, forming pellet 1, as shown in the diagram. This is most likely going to be the nucleus, considering it's an animal cell and the heavy structure is the nucleus. The liquid above pellet 1 was poured into the clean centrifuge tube and so we took the fluid remaining by filtering or something, and we poured it into a clean centrifuge and spun the centrifuge at a higher speed to separate the next heaviest cell structure, which is going to be most likely the mitochondrium, because we don't have any chloroplast. This cell structure sank to the bottom, forming pellet 2. Now this procedure was repeated twice more to obtain pellet 3 and 4, each containing a single type of cell structure. Which row shows the correct order in which the cell structures are collected? So I believe pellet 1 is the nucleus, pellet 2 will be the mitochondria, pellet 3 lysosomes, then ribosomes, because ribosomes are the tiniest. And I like to think about it considering this Fedberg unit here, it's like 80s or 70s, which means basically that if you think about it in terms of diameter also, that could help you out. Radioactively labeled nucleotides are introduced into a cell. In which cell structures would the radioactivity become first concentrated? Considering we're introducing nucleotides, it will be 2. Sometimes we'll ask about amino acids, so of course that should go directly into the, I'd like to say ribosomes, but it's not an option here. So I would say that the answer here is 2. In order to structure become first concentrated, it will be 2, and 3 also because there's transcription and translation happening in the mitochondria, considering it has, if you remember, circular DNA. So I would say 2 and 3, which is C. What is the general formula for amylose? Amylose is a polymer, and for all polymers they have the formula C6H10O5. So here I have a diagram to represent to you how water is being lost. I mean, you get the point. Basically, when the polymer is going to be forming, this is going to be a condensation reaction. In condensation reactions, water is lost, right? And that's why it's going to always be C6H10O5, considering there are always 6 carbon atoms in each single subunit, and we've lost 2 H for water and 1 O for water again. Now the diagram shows the relationships between some important molecules and bonds. Okay, he says which is represented by circles numbered 1, 2, and 3. So let's see. So 1 is peptide, and says bonds formed by condensation reaction. That is true. They're never formed by hydrolysis. Hydrolysis is a breaking down of bonds and not the formation of bonds. It's a breaking down of a covalent bond using water. So peptide bond, yes, it is formed by condensation. 2 is carbohydrates. This is true. 2 is carbohydrates, yes. And 3 is lipids, which is correct because vegetable oil, triglycerides, these are all lipids. Okay, so we can answer. Next. He says that in unsaturated lipid molecules, where are the double bonds located? So the double bonds are going to be located within the fatty acid chain, and it's going to be between the carbon atoms. And so that means it's within the fatty acids only. Phospholipids and triglycerides are important biological molecules. Which properties are correct for these molecules? Nonpolar phospholipids. Actually, they like to describe triglycerides as nonpolar. Partially hydrophobic, well, phospholipids because they have the hydrophobic fatty acid tails, but the hydrophilic phosphate groups. Moving on. The diagram shows 2 arrangements of amino acids in a protein. He says which row correctly names the bonds at S or T. So here we clearly have an alpha helix and a beta pleated sheet. Excuse me for the terrible handwriting. He asks what are the bonds at S and T. We know that in a secondary protein structure, which is like alpha helix or beta pleated, the bonds holding what's called coils together or the parallel lines together are going to be hydrogen bonds. Therefore, the bonds at S and T are hydrogen bonds. Ionic bonds are always absent in secondary protein structure. He says students were asked to highlight only the R groups of 2 ring-shaped amino acids, and which pair of diagrams are correct for both amino acids. The R groups are the variable side chains. So normally in an amino acid we have the alpha carbon and we have another carbon. That's where the R. So we have an alpha carbon that's part of the carboxyl group, the alpha carbon, and nitrogen, right? And then basically here we'll have the OH and double bond O, and here we're gonna have what's called R group, NH, NH, NH. And then he says which pair of diagrams are correct for both amino acids. So we're looking at something that's not here or there, it's just attached to the alpha carbon, to the central one. And so this is not exactly correct because what exactly is this? Alright, let's see. Here we have the R group. He circled the carbon of the carboxyl group. That's not right. This is also wrong, and this is also wrong, and this is right. But basically, again, some students were asked to highlight the R groups of 2 ring. Let's check out D. So yes, this is right. And why do they keep on putting this bond over here? Let's see. We have. I suppose like they do form a bond sometimes. So basically, I think this is the answer. It says which words from the table correctly complete the paragraph about enzymes. When the pH of an environment is decreased below the optimum pH of an enzyme, are the bonds between adjacent R groups, so they're ionic bonds, right? Between so hydrogen and ionic, obviously not peptide, the last to possibly break, and R groups holding the tertiary protein structure together. So that's A. Alright, the effective substrate concentration on the enzyme catalyzed reaction was measured in 3 different conditions: with no inhibitor, with a competitive inhibitor, with a non-competitive inhibitor. Since the graph shows the results. So which statement is correct? Substrate concentration, inhibitor X, inhibitor Y, enzyme with no inhibitor, and rate of reaction. So X is a competitive inhibitor which binds to the site other than the active site. That is immediately wrong, because the competitive inhibitor will bind to the active site, and that's why it's called competitive, because it will compete with the substrate for the active site of the enzyme. A non-competitive inhibitor which has a similar shape to the active site of the enzyme. Similar shape is really a tricky word. It does not necessarily have a similar shape. It actually would most likely not have a similar shape, so let's just ignore that. A competitive inhibitor has a similar shape to the active site of the enzyme. Again, another tricky word. That's the reason I'm saying this is tricky, because the inhibitor will have a complementary shape to the active site and a similar shape to the substrate binding site, right? So it's not similar to the enzyme active site, because if I said that the enzyme active site looked something like this, then the competitive inhibitor would have to have this type of shape, not this type of shape, right? So why is a competitive inhibitor why is a non-competitive inhibitor, which is true, and that's dependent on the graph, right? So what's it called? Which binds to a site other than the active site of the enzyme? Yes. Again, why is this a non-competitive inhibitor? Non-competitive, this is competitive. You're gonna say because as substrate concentration increases, it will affect the inhibitor activity. Because here, yes, it will compete with the substrate on the end. So let's say this is the competitive inhibitor and this is the substrate, right? It will compete with the substrate on the active site, but once you increase the quantity of the substrate, it will no longer be a competition. It will be like, okay, I'm gonna be here more, so might as well just bind to me, right? And that's basically what happens when it comes to this. As you can see, it will increase. It's a lower rate of reaction, but eventually it will increase to reach the Vmax, the maximum possible rate of reaction. For inhibitor Y, it will decrease the Vmax, but it will not change the affinity of the enzyme to substrate. Okay, next question. So which statements about the cell surface membrane are correct? Channel proteins allow water-soluble ions and molecules across the membrane. That is true. Glucose can pass into the cell via carrier proteins or channel proteins, yes. And oxygen passes freely through the membrane as it's soluble in lipids. That is true. And some glycoproteins act as antigens. That is also correct. So that is A. So which of these features increase the efficiency of ion uptake by root hair cell? So specifically, the root hair cell has lots of adaptations, right? It has a large surface area. It has a large vacuole with a concentrated cell sap, right? But let's see what is exactly important for increasing the efficiency of ion uptake. Many mitochondria are perfect because we know that the ions are taken up by the cell by active transport, which is an active process, as like from its name we can tell that, right? And high concentration of ions in the vacuole. This is not really important because active transport doesn't really rely on the presence of a concentration gradient, and protein carriers in the cell surface membrane, obviously, because active transport relies on these. What can increase the fluidity of the cell surface membrane at low temperatures? Double bonds between carbon atoms and fatty acid chains, cholesterol, fatty acids having shorter chains. The truth is, all of them, because double bonds between carbon atoms and fatty acid chains do increase fluidity. The presence of shorter fatty acid chains will increase the fluidity, and cholesterol, one of its roles is to regulate the fluidity. At low temperatures, it will increase the fluidity. At higher temperatures, it will decrease the fluidity, and that's basically by preventing the fatty acid chains from moving a lot and moving away from each other. It will push them towards each other, and that's how it will decrease the fluidity and increase it at lower temperatures. And so the answer is A. Now, this is an important point. Don't forget that actually they could get this question but reversed. So he says, what decreases the fluidity of the cell surface membrane at high temperatures? So here you'd say the absence of double bonds, or let's say the presence of more saturated fatty acids or fatty acid chains, because saturation means no double bonds. Non-unsaturated means at least one or more double bonds, okay, between the carbon atoms, okay. So of course, basically, what will decrease the fluidity in high temperatures, as I mentioned, the presence of saturated fatty acids only, and longer fatty acid chains, and of course cholesterol. Okay, giving this question a read. Obviously here they're asking for surface area to volume ratio. And this basically like discussing, because as you can see they have all the labels for the length, width and height. Now here I just want to establish what are the formulas. Volume equals to length by width by height for a cuboid, or for for a cuboid, okay. And surface area will equal to 2 multiplied by the length by the width, 2 multiplied by the height by the length, 2 multiplied by the height by the width. So if we calculate here, they already gave you surface area, but they didn't give you the volume, which we will calculate, and we find it to be 5,000 micrometers cubed. Here, if you want to calculate surface area, you will find it to be 1450 micrometers squared. So which statement is correct about the palisade cell and epithelial cell as shown in the diagram? So an increase in surface area reduces the distance for gases to reach center. Actually, no, not necessarily, and maybe even wrong. Okay, surface area of the palisade mesophyll cell is 500 times greater than the columnar epithelial cell. So we can see the surface area is actually smaller for the palisade cell, the mesophyll cell, compared to the columnar. The surface area it's greater, but it's no way greater by 500 times. Let's check using the calculator in case we were terrible at math. I mean, sometimes I need to check for 1 plus 1, but so I have to check. It's 1.51 times greater, so no, it's not 500 times. The surface area to volume ratio is greater in the columnar epithelial cell than the palisade mesophyll cell. Let's calculate surface area to volume ratio, and that's simply by dividing the surface area to the volume. Now remember to never judge how big the surface area to volume ratio is compared to another cell unless you have it in like decimal form, because sometimes you would get confused and you would not know which one is larger, which one is smaller. And so I always like to convert my fraction to a decimal in order to see which one is actually greater. So in this case, if I calculated the surface area to volume ratio for the palisade mesophyll cell, it will be 0.44. What's it called? If you divide 1450 by 2250, it should be 0.64. So which one has a greater surface area to volume ratio? It is greater in the columnar epithelial cell. That's true. Well, a student observed cells in the growing region of an onion root and obtained the data shown. These are the stages, and this is the number of cells. So what percentage of cells contains chromosomes that appear as 2 chromatids? So chromosomes that appear as 2 chromatids have part of interphase all the way until anaphase, but like anaphase we cannot include this. So I can either include from here to there, right, or basically including metaphase, but interphase, but I'm not going to do that because what's it called? He wants percentage of cells containing chromosomes that appear as 2 chromatids for the majority of this period. Not all of them are going to have that. So I think we can just calculate it by 73 plus 16 divide 886 plus 14 plus 73 plus 16 plus 11 times 100. It will be 8.9%, which is B. Moving on, we have which of these events are part of mitosis. Now, mitosis is different from the cell cycle because the cell cycle includes everything, starting from a G1 to S phase to the G2, meaning the interphase, and then what you call the mitotic cell cycle, and then cytokinesis. But the mitosis or the mitotic cell cycle includes only PMAT, being prophase, metaphase, anaphase, telophase. So again, if I draw this right here, this is interphase, this is mitosis, this is cytokinesis. The cell cycle is from here to there. That's the cell cycle. This is the mitotic cell cycle. In other words, it's mitosis. So mitosis will only include telophase. That's D. Okay. Now, during which phase of the cell cycles does DNA replication take place? During synthesis phase, hence the name. Now, which row is correct for guanine? Guanine is a purine, that is correct. It has a double ring structure, that is why it is a purine, because it has a double ring. Okay. Joins to its complementary base with 3 hydrogen bonds, yes, and the complementary base is cytosine. So A. Moving on, and Stahl. These are the scientists that investigated DNA in bacteria, and they grew bacteria in the medium with only heavy nitrogen N15 until all the bacterial DNA was heavy. Now, these bacteria moved from a heavy nitrogen medium and cultured with only light nitrogen N14, and a sample of bacteria was collected from the first generation and in the medium containing light nitrogen, and their DNA was analyzed. So again, if we understand what's going on here, we have I think the medium here is cesium chloride. This is the medium we put DNA mostly in. And when you place the DNA in here, right, it's gonna undergo semi-conservative DNA replication. And they left it to divide until like all its DNA, right, became made of this N15 like nucleotides, because nucleotides contain nitrogenous bases. Nitrogenous bases are gonna be made from this heavy nitrogen, right? So basically, we're gonna end up with this type of heavy nitrogen, and then basically they took this DNA, they took the DNA right here, and they placed it in a medium containing N14, okay? And they left it to divide, so here we have N14. And basically they left it to divide from the first generation Yes, and basically hybrid DNA contains, and this was analyzed, and it was hybrid. Look at this, like here, let's assume that N14 is a red medium, and that this was the purple medium. Okay? Then basically, when they left it, they have hybrids. So one DNA strand was N15, and one was made of N14, and one again made of N15, and so on. So 100% was hybrid. The new strand is going to be complementary to the old strands, will be synthesized from the N14. So which row shows the percentage heavy DNA strands and percentage of hybrid DNA strand molecules in the first generation produced in a medium containing light nitrogen? Again, there will be 100% hybrid as I have shown here, but the percentage of heavy strands will be 50% because, as you can see, there are 4 strands, for example, 4 DNA, 2 DNA molecules, 4 strands. 2 of them are N14, and it will basically being the light strands, and 2 of them N15 being the heavy strands. Therefore, it's 2 out of 4, which is half, 50%. Okay, moving on. Now, which is correct? The correct DNA triplet on the original DNA template that codes for the amino acid histidine. So histidine over here shows me the anticodon. Now this is a nice trick. If you want to convert from anticodon to code, which is a triplet basis for DNA, you just have to change any U's into T's, right? Because uracil is going to be in case of like RNA, but we don't have uracil in DNA. So that means basically that you have to convert the U over here to a T. And that is for DNA code. Again, if he said that there's an anticodon, just immediately change the U into a T to find the code in DNA. In case it's a codon, it's a totally different case. The photomicrograph shows tissues in a stained transverse section of a plant stem. Students were asked to draw 4 adjacent xylem vessel elements, which drawing all are all drawn to the same scale is correct. Okay, they're all drawn to the same scale. Which drawing is right, knowing that they're meant to be drawn, I think, to this scale, and which one looks like the xylem. And we know that the xylem is right here. Hold on, this is the xylem, okay? And this is the sclerenchyma, I guess. And here we have the phloem. So basically, because this is a stem, yes, so like you would imagine that this is the sclerenchyma, followed by the phloem, followed by the xylem. This is the ugliest drawing I've ever made, but it doesn't matter. I suppose basically that the cells have some type of thick cell wall as is drawn here, and they have some type of pentagonal and hexagonal shapes, so I would say the answer is D. Oh, sorry about that. Okay, yes, moving on. What is the function of hairs, trichomes, on xerophytic leaves? So adding waterproof layer? No. Protecting the stomata? No. Reducing the surface area? No. Trapping a layer of moist air? Exactly. And that is to reduce the water potential gradient and hence of water vapor, of course, and hence reducing the rate of diffusion of water vapor out of the leaves, and hence reducing the rate of transpiration. So saving water and preventing water loss and preventing wilting. Now in this question, which features of companion cells are essential for their function? Now they're connected to plasmid to this by plasmid to the sieve tube elements. That is true, and it's essential because it's needed for loading when sucrose and amino acids are going to move through the symplastic pathway, in other words through the plasmodesmata from the companion cells into the sieve tube elements. They have a thinner cell wall compared to the sieve tube element. Now this is not exactly what I was wanting to hear if I wanted this to be an essential feature to their function, because yes, companion cells do have a thinner cell wall, but it's only at the region where it is in common between the companion cell and the sieve tube element. So this region will have an extremely thin cell wall, but the other regions will actually have a thick cell wall, and the reason for this is we want to reduce the diffusion distance, but on the other side we don't have such a thin cell wall, so we can't say that it has a thinner cell wall overall because we don't really know that. Okay, so I would not say this is a feature that contributes to its function. They contain a nucleus and a mitochondria, then of course, because mitochondria are responsible to provide us ATP, which is going to be hydrolyzed to release energy. Energy is required for the loading. It's an active process. I mean, like in general it will be required for loading, but specifically it's required for active transport of hydrogen ions out of the cytoplasm and into the cell wall or the surroundings. And of course the nucleus is required for lots of things: transcription, for protein synthesis, to synthesize the protein carriers required for the pumping here, the ATP powered proton pumps, I believe. Now and hence the answer is C. Shows a photomicrograph and shows a human blood smear. So which row identifies labeled cells? So let's identify them together. This is a monocyte. And because this is a slide in the blood, I would call it a monocyte and not a macrophage. For 2, this is a neutrophil, and I can tell by its lobed nucleus. Okay, sorry. And 3 is a lymphocyte, and I can tell by how the nucleus occupies almost all of the cell. And this is a repeated question. It's a lovely question. So which row identifies labeled cells? Let's see. Again, 1 monocyte, 2 neutrophil, 3 lymphocyte. Perfect. Moving on. Now, the diagram shows the changes in electrical activity in the human heart muscle during one cardiac cycle. This is called electrocardiograph and ECG. So, stenotic node stimulated and stimulates ventricular systole. Okay, so which electrocardiograph shows a rate of 75 beats per minute? 75 beats per minute will be happening when basically, so let's see, this is 1 heartbeat from here to there. Here we have 1, 2, 3 heartbeats in 4 seconds. If you divide 3 by, if you see basically like, the rate is 3 out of 4, and that's basically just in 4 seconds. So in 4 seconds there's 3 heartbeats. In minute, which is 60 seconds, how many heartbeats will you have? Let's see. Is if it's 30? No, it says 45. Now here we have 1, 2, 3, 4, 5 beats in 4 seconds. Again, in 60, how much will you have? Cross multiply 5 by 60, divide by 4, 75. This is my answer. Which effect could be due to the reduced concentration of carbonic anhydrase? So let's see. Carbaminohemoglobin concentrations will decrease. This has nothing to do with the function of carbonic anhydrase. Carbonic anhydrase is an enzyme with a really high turnover rate. It catalyzes the conversion of carbon dioxide and water into carbonic acid, which is a weak acid. It's H2CO3, and here is carbonic anhydrase. Carbaminohemoglobin forms when carbon dioxide would bind to the terminal amine group of hemoglobin. So if you imagine this is a hemoglobin and there's some type of amine group, and then carbon dioxide is going to come and he's going to bind. It has nothing to do with carbonic anhydrase. Less oxygen is released from oxyhemoglobin for active tissues. Less oxygen is released. I mean, I could say so. Yes, I could say so. Okay, and the pH of the blood will be lowered. Actually, no, because if you have a reduced concentration of carbonic anhydrase, you would have less carbonic acid produced. Therefore, there will be less dissociation of carbonic acid in order to form hydrogen ions, which will lower down the pH, right? And now the pH of the blood will be lowered, again wrong, and the rate of dissociation of carbonic acid is increased. Actually no, because you have less carbonic acid, therefore there will be less carbonic acid dissociating. Therefore the only answer here is this one. And if you want an explanation, less oxygen is released from the oxyhemoglobin for active tissues. If you have less hydrogen ions, then hemoglobin will have a lower affinity to the hydrogen ions. Therefore hemoglobin will bind to, the oxyhemoglobin will bind to hydrogen ions at a lower rate. Therefore there will be a reduced rate of dissociation. This is known as Bohr's effect and the buffer action of hemoglobin. If you want to review this, then there are lots of notes online. I'm going to just quickly draw this for you. This is oxyhemoglobin. It will bind usually to hydrogen ions in case there's a high concentration of hydrogen ions, like in the region of respiring tissues, right? Because basically of the stuff that you saw right here. Because hemoglobin will have a higher affinity to bind to it, and so it will form hemoglobinic acid and 4 molecules of oxygen, which are released to the muscles. If you have less of like the hydrogen ions, considering there's less carbonic acid, considering there's less carbonic anhydrase, then you of course will have less oxygen. It's really cool to understand this pathway, and I hope this helps you understand it a little bit. Which statement correctly compares blood plasma and tissue fluid in a healthy person? Blood plasma contains more protein than tissue fluid. Actually, yes, that makes sense, because blood plasma consists of both like the large size plasma proteins and the small size plasma proteins, whereas tissue fluid is only composed of the small size plasma proteins. Both blood plasma and tissue fluid contain red blood cells. Actually, neither of them contain it, because blood is consistent of blood plasma and red blood cells, meaning red blood cells are not part of the blood plasma, which is a common mistake people do, thinking that red blood cells are part of the blood plasma. Tissue fluid contains white blood cells, whereas blood plasma does not. Actually, both of them contain it. And tissue fluid is formed from blood plasma and does not return to the blood plasma. No, it does return. Therefore, the answer is A. Hold on. The diagram shows three features found in tissues of the gas exchange system. So which tubes of the gas exchange system could be presented at position P in the diagram? We have selected epithelium, we have goblet cells, we have cartilage. So who contains all this stuff? Trachea and bronchus, but never the bronchioles. Bronchioles don't contain any cartilage. Okay. Moving on. The diagram shows a magnified section of part of the lungs containing specialized tissues. So which row is correct for structures labeled 1 to 6? Okay, so 1 is, as you can see, the macrophage, and it's hanging out here in the alveolus. And then we have 5 being the blood plasma, 3 being their blood cells, you can see by its biconcave shape, and 2 being this endothelial cell of the blood capillary. And let's see the answers here. So what containing high proportion of carbonic anhydride, obviously their blood cells, so that's 3. And then hydrogen carbonate ions will be obviously the blood plasma because of, the chloride shift and everything. And then lysozymes, it's not really the chloride shift, hydrogen carbonate ions will diffuse by facet diffusion out of their blood cell into the blood plasma during Bohr's effect. Lysosomes will be found also, I think, so yeah, 6, obviously, because you have phagocytosis happening, and lysosomes have a big role in phagocytosis. Moving on. So a short-term effect of smoking. Smoking is no longer part of the syllabus, so we can move on. So the symptoms, I think it's another question that's not included. So here. What statements about non-infectious diseases may be correct? It can result from a mutation. Yes, like in the case of cancer, like in the case of sickle cell anemia, these are non-infectious, yet they are resulting from a mutation. It can be transmitted by an insect vector. No, it is infectious diseases that can be transmitted. In fact, non-infectious diseases, their definition is literally a disease which is not basically transmissible, meaning it cannot be transmitted from one person to another, and diseases that are not caused by pathogens. So it can be transmitted from mother to child. Again, not transmissible. But there's no such option. So let's see. It can be transmitted from mother to child. I suppose they mean by it could be inherited from the child. So yes, okay, 1 and 3, C. Now the diagram shows one way of testing the effect of an antibiotic on bacteria. He says the table shows the results of testing 5 different types of antibiotics. Okay, here we have the table, and we have a petri dish with a nutrient agar containing bacteria, colonies of bacteria, and a disc filter paper soaked in antibiotics. So the antibiotic is going to diffuse and kill the bacteria if they are sensitive to it and not resistant. So for 5 days, and you can see that it actually did, because here we don't have any of these spots. Sometimes they would say, sometimes they would show this as a gray part, not necessarily spots, but this also helps us to understand, so that's great. Again, zones of less than 30 millimeters show the presence of resistant bacteria. Okay, so if it's less than 30 millimeters, that means that bacteria are resistant. Okay. Let's see. So basically we could see that for day 1, for type 1 bacteria, you could see that it's 24 and it does not reach 30, meaning these bacteria are not resistant. Here, the bacteria are most definitely resistant because the zones are less than 30 millimeters. And here the bacteria are also resistant, and here the bacteria here also resistant. So bacteria types 1 and 5 are not resistant and are sensitive to the antibiotics. So all the types of bacteria become resistant to the antibiotics over time? No, as we can see. And it's not a conclusion that we can support by this data. Only types 2, 3, and 4 of the bacteria show resistance to the antibiotic. That is true. And the antibiotic can be used to treat types 1 and 3? No, because 1, yes, it is sensitive, so yes, I can use it to treat bacteria 1, but I cannot use it to treat the resistant type 3, of course. The type 5 of bacteria can never become resistant to the antibiotic. No one said so, okay. So what's it called? 37. Some animals have genes that code for small peptides called cathelicidins. Okay. And these peptides kill a wide range of bacteria by attaching to lipids in the bacterial membrane. So we can disrupting them. So I suppose these are some chemicals with bactericidal effects, or we can say they're antibiotics. Now, some scientists have produced a synthetic version of this thing that kills bacteria that are resistant to a number of antibiotics, such as tetracycline. So which pair of statements explains how the synthetic cathelicidin might help with the problem of antibiotic resistance? It's synthetic, so bacteria can never become resistant to it. No, okay. It could be used instead of tetracycline, allowing tetracycline resistance to be reduced. It will not reduce tetracycline resistance, it will just, actually that makes sense, yes, because like if people ditch, or let's say people leave tetracycline for a little bit because they use the other chemical, then it makes sense for bacteria to become more sensitive to tetracycline. The only way a bacterium, because like there's less of a selection pressure, the only way of a bacterium could develop resistance to it is by altering all the lipids in its membranes. No, it's just by altering a couple of nucleotides and a mutation. It could be used to kill multi-drug resistant strains of bacteria for which there is no effective antibiotic. I suppose yes, because the bacteria is still not resistant to it since it's still brand new, and I suppose. So I could say 2 and 4, okay? Now, moving on, he says that hybridomas are used as a basis for the production of large numbers of monoclonal antibodies, which statement describes how hybridomas are made. So it's fusing, this is what actually happens. The plasma cells are going to be fused with B myeloma cells, in other words, cancer cells. And here he says that we fuse B lymphocytes. Sometimes they like to say B lymphocytes instead of plasma cells, but yes, okay. Which statements about the properties of antigen binding sites in an antibody molecule is correct. They are located on the light chains only. Actually, no. The antigen binding site is this part. So if I were to draw this, it's part of the heavy and light chain. It's a variable region here. So, no. Okay. They have a hinge region to give flexibility for different antigens. Now, antigen binding sites do not have the hinge region. This is the hinge region. Okay, so I would say the antibody itself has the hinge region, but I would not say this answer is right. They have binding sites for receptors on phagocytes. But again, he says the properties of antigen binding sites, so that is also wrong. Okay, because this is the concentration and this is the binding site for phagocytes. Now they have a variable amino acid sequence for different antigens, exactly, because they have different protein structure, hence they have different secondary and tertiary and quaternary protein structures, and that's why they are variable regions and variable amino acids and variable everything. Okay, last question. The statements describe ways in which different types of monoclonal antibodies can work. Binding to proteins on cell surfaces and triggering the immune system. Okay, binding to proteins on this and triggering that is one way, like in the treatment of cancer. Blocking molecules on cell surface membranes that inhibit T cells. So blocking molecules that inhibit T cells, that makes sense. Blocking cell signaling receptors that trigger cell division. Yes, we'd say cell division because there's continuous and uncontrollable cell division in case of cancer. That's what results in the formation of tumors, and we want to stop that, and that's how we use antibodies to do that, right? So 3, I think, is right. So, blocking the signaling receptors that trigger the immune response. No, actually want the immune response to happen, so I believe B.